Okay, so good evening everyone. Today is the fourth live session of the series of classes, live sessions basically of this course, Host Pathogen Interaction Immunology, taken by course instructor Professor Himanshu Kumar from IISA Bhopal. So what we do in these live sessions is that we discuss some previous year old assignment questions, and we not only discuss the answers to those particular questions, but we also try to discuss the concepts behind those questions so that it's easier for us to do similar type of other questions, right? And we are clear with the concepts. Okay, so that is the basic gist of why we do these kinds of live sessions. So my name is Aishi. I'm a PMRF research scholar from the Department of Chemistry, IIT Bombay. And now in these live sessions, we try to keep this as interactive as possible. So I say the question, I'll wait for a few minutes and I'll let you answer. Then I'll move ahead and discuss the questions. At any point, if you feel like you want to interrupt or you have any query, any doubt, even after the discussion ends, I'll wait for a few minutes before moving on to the next question. So if you have any doubt, you can just unmute your mic and let me know, or you can write in the chat box. Anything is fine. If I know the answer at the moment, I'll tell you. If I don't, then you'll have to give me some time. I'll read it and get back to you by next class, right? So that is how we conduct these live sessions. So with this in mind, let's move on to the first question for today. What is the role of GM-CSF in immune cell development? First, you have to tell me the full form of GM-CSF. Options are A, it stimulates the production of antibodies. Option B, it causes hematopoietic stem cells to differentiate into dendritic cells. Option C is it inhibits T cell proliferation. Option D is it acts as a hormone that regulates glucose levels. And option E is it promotes wound healing. Which one do you think is the correct answer? I'll wait for a few minutes, then you can, and I'll wait for you to tell me the answer, then I'll move ahead. So I'll repeat the question again. What is the role of GM-CSF in immune cell development? Option A, it stimulates the production of antibodies. Option B, it causes hematopoietic stem cells to differentiate into dendritic cells. Option C, it inhibits T cell proliferation. Option D, it acts as a hormone that regulates glucose levels. And option E, it promotes wound healing. So you have to give me the answer in detail and also have to tell me the full form of GM-CSF with regards to immunology. I'll wait for a few minutes. Okay, so basically the answer will be option B, which is it causes the hematopoietic stem cells to differentiate into dendritic cells. So the full form of GM-CSF pertaining to immunology is granulocyte macrophage colony stimulating factor. Okay. Now, granulocyte macrophage colony stimulating factor is a cytokine that plays a central role in hematopoiesis and immune system development. It primarily acts on hematopoietic stem and progenitor cells in the bone marrow. Now, it stimulates differentiation of progenitor cells into granulocytes like neutrophils, eosinophils, monocytes or macrophages, and dendritic cells, especially important for antigen presentation. It enhances survival, proliferation, and activation of these immune cells. Now, it boosts functional activity of phagocytosis antigen presentation as well. So, in laboratory and immunological context, GM-CSF is widely used to operate to generate dendritic cells from bone marrow or monocytes, which directly supports option B over here. Why the other options are not correct? Option A, it stimulates the production of antibody. This is mainly the role of cytokines like interleukin 4, interleukin 6 and interleukin 21, not GM-CSF. Option C is not correct because it inhibits T cell proliferation. GM-CSF is generally pro-inflammatory and stimulatory, not inhibitory. Option D, which regulates glucose levels, it is the role of hormones like insulin, glucagon, not cytokines like GM-CSF. Option E promotes wound healing. So while GM-CSF can indirectly aid tissue repair by immune activation, this is not its primary or defining function. So that is also not correct. Now we have talked about the evidence of decline in immune function with aging. Like as we have talked about, aged individuals have increased incidence of infections like pneumonia, influenza, tuberculosis, meningitis, urinary tract infections. Increased incidence of autoimmune diseases like rheumatoid arthritis, lupus, hepatitis, thyroiditis, Graves, hyper Hashimoto's, multiple sclerosis of obviously. They have increased cancer incidence like prostate, breast, lung, throat, neck, head, stomach, colon, bladder, skin, leukemia, pancreatic. Then there is tolerance to organ transplants like kidney, skin, bone marrow, hearts, liver. All of these decreases. Now the function of immune system is protection against bacteria, virus, fungus, cancer, toxins, and any 5000 up Dalton substances like protein, lipids, nucleic acids. Now, obviously, cells derived from stem cells, liver and bone marrow, the cells are stored, multiply, interact and mature in thymus, spleen, lymph nodes and blood. Accessory organs can be appendix, tonsils, intestines. We have discussed about this. We have discussed about B cells also. I am not going into too much detail. Okay, so if you can see here, these are chemical mediators or modulators, cytokines basically. They influence proliferation, differentiation and survival of lymphoid cells, as numerous actions on other body cells can compromise the following, like interleukin family. There are 16 different proteins from IL-1 and up, and numerous effects on lymphocytes and other cells with IL receptors. Then there is granulocyte macrophage colony stimulating factor GM-CSF. It regulates hematopoiesis, affects phagocyte function and angiogenesis. Then we have colony stimulating factor. It's basically a glycoprotein regulating white blood cell production, activity and survival. Then we have tumor necrosis factor TNF alpha, cytotoxic against malignant and inflammatory cells. produced primarily by macrophages. TNF-beta, cytotoxic against malignant cells, enhances phagocytosis, produced primarily by T cells. If we talk about interferons, interferon B, produced by many cells, antiviral actions, interferon gamma, synthesized by activated NK and T cells, involved in the activation of macrophages and inflammation. Okay. So you can go through this paper, which is basically published in Journal of Allergy and Clinical Immunology. GM-CSF along with IL-4 but not alone is indispensable for differentiation of human dendritic cells from monocytes. Okay. Not going into too much details. Okay. Now if we talk about cancer and the immune systems, we talk about tumors and metastasis. Cancer is basically altered cells that have escaped normal growth regulation mechanisms. Neoplasm tumor can be benign or malignant. Metastasis is basically spreading of cancerous cells via blood or lymph to various tissues. Okay. Carcinoma is basically endodermal or ectodermal tissue. Leukemia is hematopoietic stem cells or lymphoma. Sarcoma will be mesodermal connective tissues. Now, what makes cancer exactly? It's the decreased requirements for growth factors in serum. They are no longer anchorage dependent and they grow independently of density. So, normal cells eventually enter G0. They can confluence monolayer, they can contact inhibition, they can perform contact inhibition. But basically, this all leads to checkpoint failure and that leads to cancerous cells. Now, what is the malignant transformation? They are like in vitro cancers. Like there can be two phases. Initiation, that is changes in genome. Promotion, that is proliferation. Okay. So, basically what happens is that it can be because of chemical and physical carcinogens, virally induced transformation, cultured tumors, good models for study that we do in biolabs, and cancer cells are basically immortal. So, oncogene is cancer gene, often found in viral genomes. Proto-oncogene is cellular counterpart, can be turned into an oncogene. Okay. So, induction of what can go right? So, induction of cellular proliferation, inhibition of cellular proliferation, tumor suppressor genes, and regulation of programmed cell death. And what can go wrong? Chromosomal translocations, tandem repeats, mutations in proto-oncogenes, viral integration, growth factors, and their receptors, all of this. So, what is the process of induction of cancer? There are proto-oncogenes, the expression of which is essential for growth controlling proteins like growth factors, growth factor receptors, signal transducers, intranuclear factors, regulators of programmed cell death, and all of that. Then can be undergoing retroviral transduction to lead to transformed cells or viral oncogenes. Or they can undergo mutagens, viruses, and radiation. They can be exposed to all these and genetic predisposition, obviously, and that can lead to cellular oncogenes, the expression of which will be qualitatively altered hyperactive proteins, quantitative alterations like gene amplification or translocation, resulting in increased or decreased levels of products. Now, lymphomas, solid tumors with lymphoid tissue like bone marrow, lymph nodes, thymus, Hodgkin's and non-Hodgkin's. Leukemias proliferate as single cells, acute or chronic depending on the progression of the disease. Acute, they can appear suddenly and progress rapidly, arise and is less mature. Chronic is much less aggressive and develops slowly, mature cells. TSTs are tumor-specific transplantation antigen. Okay, so they are uniquely tumor cells that do not occur in normal cells in the body. They are novel proteins treated by mutation present in a class 1 major histocompatibility complex. They can either be chemically, physically induced or virally induced tumor antigens. Now, if you talk about chemically, physically induced, they can be specific immunological response that can protect against later challenge by live cells of the same line, but not other tumor line cells like methylcolanthrin or uvealide. Virally induced can be expressed tumor antigens shared by all tumors induced by same virus like Burkitt's lymphoma, Epstein-Barr, HPV, etc. These are not unique to tumor cells. They occur in nervous cells in the body. So what's the problem? Like fetal or adult presence, concentration of growth factors and growth factor receptors. So if we talk about virally induced tumors, they have the same antigens for each tumor caused by that virus, like HPV. And what is the immune response to tumors? Mostly cell-mediated response, like natural killer cells. They are not MHC restricted. So the Fc receptor binds to antibody-coated tumor cell. Okay, and macrophages, they are also not MHC restricted. Elicits ADCC and TNF alpha. Immune surveillance theory. Now, if you ask me about what is ADCC in the immunology concept, I will have to tell you that we have already discussed in one of the previous class, but basically it stands for antibody dependent cellular cytotoxicity. Right? It's an immune defense mechanism where immune cells kill a target cell that has been tagged by or opsonized by antibodies. So how ADCC works? Antibodies bind to a target cell, typically a virus infected cell, tumor cell or parasite. Antibodies, usually IgG, attach via their FAB region to antigens of the target. Affected immune cells recognize the antibody-coated cell, immune cells like natural killer cells, macrophages, neutrophils. They bind via Fc receptors to the Fc region of the antibody. Then we have activation of the effector cells. Binding triggers activation of the effector cell. Then target cell killing. NK cells release perforin, form spores and granzymes that induce apoptosis. It leads to death of the antigen-coated and target cell. Antibody-coated target cell. So basically, the ternary complex and co-stimulation by B7 will happen in case of cancer. They can activate lymphocytes, release IL-2, interferon gamma, TNF alpha, and cancer cells must be susceptible to killing. CTL lysis macrophages. But if the body has all these defenses, why do people still have cancer? Bad antibodies. Some antibodies do not protect against tumor growth but also enhance it. Release of immunosuppressive cytokines like transforming growth factor beta, interleukin 10 and vascular endothelial growth factor, and hide and go kind of antigen. Antigens usually seem to hide in the presence of antibody. Also, some cancers are completely shed of the antigen. Okay. So, conducting cancer gratitude is like reduction in class 1 MHC molecules, lack of co-stimulatory signals. All of these can lead to cancer. If you talk about cancer immunotherapy, manipulation of the co-stimulatory signal, enhancement of APC or antigen presenting cells activity, cytokine therapy, monoclonal antibodies and cancer vaccines. So what is the manipulation of the co-stimulatory signal? Now tumor immunity can be enhanced by providing the co-stimulatory signal necessary for the activation of the CTL precursors. Now, it is the basis for vaccines. It prevents metastasis after surgical removal or primary melanoma in human patients. Now, GM-CSF, it is granulocyte macrophage colony stimulating factor, as they are cytokines that induce the formation of distinct hematopoietic stem lines. Now, cytokine therapy can basically use recombinant cytokines singly or in combination to augment an immune response against cancer. By isolation and cloning of various cytokine genes such as IFN alpha, beta and gamma, interleukin 1, 2, 4, 5, 12 and GM-CSF and tumor necrosis factor. Now interferons, most clinical trials involve interferon alpha has been shown to induce tumor regression in hematopoietic, hematologic malignancies like leukemias, lymphomas, melanomas and breast cancer. All types of IFN increase MHC1 expression. IFN gamma also has been shown to increase MHC2 expression on macrophages and increase activity of TC cells, macrophages and NKs. Tumor necrosis factors kill some tumor cells, reduces proliferation of tumor cells without affecting normal cells. Now, hemorrhagic necrosis and regression inhibits tumor-induced vascularization and damaging vascular endothelium. Lymphocytes are activated against tumor antigens in vitro. They are cultured with X-radiated tumor cells in presence of IL-2. They generate lymphokine-activated killer cells which kill tumor cells without affecting normal cells. Now, tumors also contain lymphocytes that have infiltrated tumor and act in anti-tumor response. Via biopsy, obtained cells into an expanded population in vitro with generated tumor-infiltrating lymphocytes. Now, if we talk about monoclonal antibodies, we have anti-idiotype, growth factors, immunotoxins, all of these. Okay. Cancer vaccines from normal to abnormal. We have talked about antigen-antibody reaction basically. It's a chemical reaction between antibodies produced by the B cells of the white blood cells and antigens during immune reaction, and the antigens and antibodies combined by a process called agglutination. It's the body's fundamental reaction by which it is protected from complex foreign molecules such as pathogens and the chemical toxins. In the blood, the antigens specifically and with high affinity bound by antibodies to form an antigen-antibody complex, and the immune system is then transported to cellular systems where it can be destroyed or deactivated. Now, there are several types of antibodies and antigens, and each antibody can bind only to a specific antigen. The specificity of the binding is due to the specific chemical constitution of each antibody, and the antigen determinant of epitope is recognized by the paratope of the antibody situated at the variable region of the polypeptide chain. Now, the variable region in turn has hypervariable regions which are unique amino acid sequences in each antibody. Antigens are bound to antibodies through weak and non-covalent interactions such as electrostatic interactions, hydrogen bonds, van der Waals forces, and hydrophobic interactions. Now, what are the general properties of antibody-antigen reactions? The antigen-antibody reaction involves specific interaction between the epitope of an antigen with the corresponding paratope of its homologous antibody. Its specificity basically. Then if we come to strength, the strength or the firmness of the association is influenced by the affinity and the avidity of the antigen-antibody interaction. Then if we come to affinity, it refers to the sum total of non-covalent interactions between single epitope of an antigen with its corresponding paratope present on antibody. Then if we talk about avidity, it's a term used to describe the affinities of all binding sites when multivalent antibody reacts with a complex antigen carrying multiple epitopes. Okay. So basically, cross-reactivity can happen in antiserum raised against an antigen can also react with a similar antigen of another type. This is called cross-reaction, and the antigen which produces the cross-reaction is called cross-reactive antigen. Now, the strength of antibody raised against its own antigen is strong. Now, the principles of specificity and cross-reactivity of the antigen-antibody interaction are useful in clinical laboratories for diagnostic purposes. One basic application is the determination of the ABO blood group. It is also used as a molecular technique for infection with different pathogens such as HIV, microbes, and helminth parasites. Okay, so soluble antigens. One second. Soluble antigens combined with soluble antibodies in presence of electrolyte at suitable temperature and pH to form insoluble visible complex. And this is called a precipitation reaction. It is used for qualitative and quantitative determination of both antigen and antibody. And it involves the reaction of soluble antigen with soluble antibodies to form large interdocking aggregates called lattice. Now, the antigen and antibody rapidly form antigen-antibody complexes within few seconds, and this is followed by slower reaction in which the antibody-antigen complexes forms lattices that precipitate from the solution. Okay, we will not talk about these in details. Okay, now what is direct agglutination? When a particular antigen is mixed with a specific antibody, agglutination occurs directly. Okay, if you can see that. That's the basis of blood group determination. We'll not go into too much detail because that is not the basis of our question. So coming back to the question that we started with, what is the role of GM-CSF in immune cell development? It causes hematopoietic stem cells to differentiate into dendritic cells. That is the correct answer. I'll wait for a few minutes. If any of you have any doubt, you can ask me. If you have any queries, you can ask me now. Then we move on to the next question for today. Okay, if no doubts, then we'll move on to the next question for today. The next question is, one sec, let me check. Okay, so the next question is, which of the following are considered pro-inflammatory cytokines? Select all that apply. So it is a multiple select question. The options are A. IL-6, option B. IL-10, option C. TNF alpha, option D. IL-1 beta, option E. MCP-1, option E. IL-4, option F. GM-CSF, option H. TGF beta. Which one do you think is the correct answer? And which ones do you think? I'll wait for a few minutes till you come up with the answer, then we'll discuss. Okay, so the correct answer will be these. Okay, so the right options will be IL-6, TNF alpha, IL-1 beta, MCP1 and GM-CSF. Okay, so if we talk about pro-inflammatory cytokines, IL-6 is one. It's a strong pro-inflammatory cytokine. It induces acute phase response. It promotes fever and immune activation. Then TNF alpha, tumor necrosis factor alpha, it's one of the most potent pro-inflammatory cytokines. It drives inflammation, fever, apoptosis. It's central in conditions like sepsis. Then IL-1 beta, that it's a classic pro-inflammatory cytokine, induces fever, leukocyte recruitment, works synergistically with TNF alpha. Then MCP1, which is a monocyte chemoattractant protein 1 pro-inflammatory cytokine, recruits monocytes macrophages to sites of inflammation. Then we have GM-CSF, granulocyte macrophage colony stimulating factor. It's a pro-inflammatory mediator, promotes differentiation and activation of myeloid cells. It enhances immune response. Now, which is strong? IL-10 is an anti-inflammatory. It's a strong anti-inflammatory cytokine. It's a wrong answer. It suppresses macrophages, dendritic cells, and cytokine production. IL-4 is anti-inflammatory. It's a Th2 cytokine. It promotes humoral immunity. It inhibits pro-inflammatory Th1 responses. And TGF-beta, which is transforming growth factor beta, is also not right because it's a potent immunosuppressive cytokine. It regulates immune tolerance, inhibits T cell and macrophage activation. Now we have talked about the lymphatic system. It drains excessive tissue fluid and not all fluid returns to bloodstream. Extra enters the lymphatic vessels. All of that we knew. Lymphatic vessels, lymphatic tissues, organs, primary or central, all of these we know. Now, lymph is a tissue fluid that has entered the lymphatic vessels, excess tissue fluid, right? So, the function is transport proteins and other molecules to the bloodstream and moves foreign particles to lymph nodes. Located behind the sternum, larger in children, site of CST cell maturation. Lymph nodes located along lymphatic pathways, absent in central nervous system. It functions like filter, remove potentially harmful particles before they enter bloodstream. Immune surveillance. Spleen located in upper abdominal cavity, similar to lymph node. Red pulp, white pulp, the site of red blood cell disposal. White pulp houses lymphocytes, site of antigen presentation. Then we talk about MALT and mucosa associated lymphatic tissue. Basically, it's a lymphatic tissue in mucosal membranes throughout the body and contains T cells, B cells, and macrophages. Can encounter antigens passing through the mucous membranes. It's found in several locations in the body: lungs, digestive tract, nasopharynx, skin, breast, salivary glands. Cells of the immune system has been broken into broad groups: myeloid, mostly in innate immune system, and lymphoid, adaptive immune system. Myeloid cells are macrophage, engulf and kill invading macroorganisms, can help induce inflammation, can activate other immune cells. Mast cells play a role in allergic responses, respond to parasitic worms, can induce inflammation. Okay, granulocyte neutrophils, most numerous, phagocytic role in bacterial infection, eosinophils and basophils. Defense against parasites, contribute to allergic responses, can be more damaging than helpful. Dendritic cells can take up and degrade pathogens. Main function is antigen presentation, activates cells of adaptive immunity. It's a bridge between innate and adaptive responses. Lymphoid cells, natural killer cells, not part of the adaptive immune system but come from lymphoid lineage. Not antigen specific, can recognize and kill some abnormal cells. Tumor cells and some viruses, thought to keep viruses at bay until adaptive response kicks in. Then B lymphocytes, antigen specific receptors, B cell receptors, once activated divides to form plasma cells. T lymphocytes in 3 categories: helper T cells provide signals which initiate response from other cells. Cytotoxic T cells kill cells which are infected with a virus or other pathogen. Regulatory T cells suppress immune activity and help control immune response. Now, the immune system is cells and biochemicals responsible for the surveillance and destruction of non-self. Innate response, quick, non-specific. Adaptive response, slower, specific. Now, chemical signaling molecules, cytokines, proteins secreted which affect behavior of nearby cells, pro-inflammatory, regulatory or growth factors, and chemokines, proteins secreted which attract cells to an area, migrate due to density gradient, and cells have receptors that bind chemokines. Now, macrophages recognize pathogens through receptors, engulf and degrade pathogens, can present antigens from the pathogen on the surface, secrete cytokines and chemokines. Inflammation recruits proteins and cells to infected areas, macrophages and neutrophils, increases lymph flow, all of these. Complement is pathogens can trigger activation of complement proteins. Activation begins a cascade which can result in destruction of the pathogen and also promote inflammation. Now, if we talk about innate immune recognition, PAMs, pathogen-associated molecular patterns, cells, macrophages, neutrophils, and dendritic cells have receptors that recognize single molecules and regular molecular patterns. Pattern recognition receptors. PAMs are present on microorganisms but not body cells. Innate and adaptive. Basically, innate response initiates adaptive response. Macrophages engulf pathogens that present antigens to lymphocytes. Dendritic cells triggered to activate T cells. Adjuvants, bacterial extracts used in vaccines, initiate an innate response which in turn activates adaptive response and leads to successful vaccination to the purified protein. Adaptive immune response initiated by APCs, primarily dendritic cells but also macrophages, engulf pathogens, degrade, then move to the antigen to the surface of the cell. Dendritic cells also have co-stimulatory molecules, stimulate T cells to proliferate and differentiate. B cells usually also require a helper T cells. It occurs in secondary lymphatic tissues and organs. Proliferation is once activated, naive lymphocytes become lymphoblasts. They divide to produce clones. Clonal expansion can divide 2 to 4 times per 24 hours for about 3 to 5 days, producing about 1000 cells specific to target antigen. These cells become effector cells, B cells, plasma cells, and T cells, cytotoxic or helper T cells. Immunological memory. Most cells generated by clonal expansion die. Some T and B cells are left behind. Memory cells reactivated quickly when the same antigen is encountered again. Provides long-lasting immunity. Now, mechanisms of adaptive immunity is cell-mediated immune response. T cells responsible for destruction of intracellular invaders. Humoral immunity, antibodies detect pathogens in blood and other extracellular fluid. Other mechanisms similar to those of innate immunity, macrophages, neutrophils complement. T cells and cell-mediated immunity, CD4 and CD8. Recognize peptides on MHC molecules, complex displayed on surface of cells, trap peptides during production and transported to surface. MHC1 protein synthesis cytosol can display viral proteins recognized by heterotoxic T cells. MHC class 2 expressed by APCs derived from proteins taken in through phagocytosis, endocytosis and can activate CD4 cells. Stimulated effector cells secrete effector molecules like mainly cytokines, recruit other effectors to target area. CD8 cytokines, cytotoxic T cells directly kill infected cells, recognize antigens. CD4 helper T cells can become different types of T cells. Now, Th1 helps control bacteria in vesicles of macrophages, stimulate them to increase intracellular destruction of bacteria. Th2 promotes response at mucosal surfaces, parasitic infections. Th17 promotes responses with loss of neutrophils, intracellular bacteria and fungi. TFH in lymphoid follicles and B cell activation. Now, if we talk about antibodies and humoral immunity, we know about antibody structures. A secreted B cell receptor, 2 variable regions can be there. Antigen binding sites and almost infinite combination of amino acids. There is a constant region, 4 to 5 forms, determines effector function, how the antibody will interact with immune cells to dispose of antigen. Okay, neutralization like binds to antigens and blocks access to cells. important in viral infections and against bacterial toxins. Opsonization is like coating pathogens with antibodies lead to phagocytosis, bacterial infections. Some evade response, innate response because they have an outer coat. Antibodies can recognize antigens on surface. Constant regions can activate the first protein, coat surface and enables phagocytosis. Antibodies also found in plasma and extracellular fluid. Antibody-mediated immunity is called humoral immunity. Body fluids used to be known as humors. Now, in primary immune response, T cells and B cells become activated for the first time. Some memory cells remain. Secondary immune response is the antigen is encoded again. The memory cells can mount a more rapid attack since they are already present. This ability can be long-lasting. If we talk about naturally acquired immunity, occurs after exposure to the antigen itself. Artificially acquired immunity occurs through the use of vaccines. Person does not become ill from the disease. And artificially acquired passive immunity, injection of gamma globulin antibodies. Naturally acquired passive immunity, antibodies are passed from mother to fetus. Okay. We have discussed about vaccines in the previous classes. I will not go into that again. Now, inflammatory mediators, TNF alpha and IL-1 are responsible for fever and the release of stress hormones like norepinephrine, vasopressin, activation of the renin, angiotensin, aldosterone system, right? So TNF alpha and IL-1 are responsible for the synthesis of IL-6, IL-8 and interferon gamma. Cytokines, especially IL-6, stimulate the release of acute phase reactants such as C-reactive protein. Other pro-inflammatory interleukins either function directly on tissue or work via secondary mediators to activate the coagulation cascade, complement cascade, and the release of nitric oxide, platelet activating factor, prostaglandins, and leukotrienes. Now, complement fragments and cytokines, like basically it stimulates chemotaxis of neutrophils, eosinophils, and monocytes. C3a, C5a increase vascular permeability. Now, cytokines, interleukins like IL-1, IL-6, and IL-8, this stimulates the chemotaxis, degranulation of neutrophils, and their phagocytic activity. They can induce extravascularization of granulocytes. Leads to fever. Tumor necrosis factor and IL-8 leukocytosis fever stimulates prostaglandins production. Now, prostaglandins are ubiquitous lipid-soluble molecules derived from arachidonic acid, a fatty acid liberated from cell membrane phospholipids through the cyclooxygenase pathway. Now, prostaglandins contribute to vasodilation, capillary permeability, and the pain and fever that accompany inflammation. The stable prostaglandins induce inflammation and potentiate the effects of histamine and other inflammatory mediators. They cause a dilation of precapillary arteries, lower the blood pressure, modulates receptor activity, and affect the phagocytic activity of leukocytes. The prostaglandin in thromboxane A2 promotes platelet aggregation and vasoconstriction. Leukotrienes are found from arachidonic acid but through the lipooxygenase pathway. Histamine and leukotrienes are complementary in action, but they have similar functions. Like histamine is produced rapidly and transiently while the more potent leukotrienes are being synthesized. Leukotrienes C4 and D4 are recognized as the primary components of the slow reacting substance of anaphylaxis that causes slow and sustained constriction of the bronchioles. The leukotrienes also have been reported to affect the permeability of the postcapillary venules, the adhesion properties of endothelial cells, and the stimulation of chemotaxis and extravascularization of neutrophils, eosinophils, and monocytes. We have talked about the. Then we talk about the platelet activating factor. Yes. Yes. Hello.
This session is about some sample question paper
Yes.
solving.
Yes.
So when will start that?
I have been discussing that only. We are discussing a cytokine question.
Ma'am, you are very fast. Can you be
Yeah.
little
I'll go
slow?
slow. Yeah. I'm discussing a question on cytokine only. Yeah, okay. So if we talk about the inflammatory mediators, platelet activating factor is also one of them. It is generated from a lipid complex stored in cell membranes. It affects a variety of cell types and induces platelet aggregation. And it also activates neutrophils and is a potent eosinophil chemoattractant. So it contributes to extravascularization of plasma proteins and so to edema. Okay. Then we will, if you see about the cellular response to this acute inflammation of the cytokines that we were talking about, it is marked by the movement of phagocytic white blood cells into the area of injury. So two types of leukocytes participate in this acute inflammatory response, the granulocytes and the monocytes. So the sequence of events in the cellular response to the inflammation includes these four steps, which is pavementing, emigration, chemotaxis, and phagocytosis. Now, if you want the meaning of emigration, chemotaxis, and phagocytosis on the basis of immunology, then these basically mean when we talk about emigration, which is also called diapedesis, it's basically movement of leukocytes out of the bloodstream into tissues. It happens during inflammation. Cells pass through the endothelial lining of blood vessels, and the key idea is basically blood to tissue movement. Chemotaxis, however, means different. It's basically directed movement of cells towards a chemical gradient. The immune cells follow signals like chemokines, IL-8, MCP1, and the key idea is movement towards attractant signals. However, this phagocytosis is different. It's basically when a natural killer cell or any immune cell, basically phagocytosis engulfs a foreign particle or an antigen, leading to its degradation. Okay. So here, pavementing basically is the release of chemical mediators like histamine, leukotrienes, and kinins and cytokines that we have been talking about that affects the endothelial cells of the capillaries, and it causes the leukocytes to increase their expression of adhesion molecules. Now, as this occurs, the leukocytes also slow their migration and begin to marginate. They move to and along the periphery of the blood vessels. If we talk about emigration, it's basically a mechanism by which the leukocytes extend pseudopodia. They pass through the capillary walls by amoeboid movement and migrate into the tissue spaces. Now, the emigration of leukocytes also may be accompanied by an escape of red blood cells or RBCs. And once they have exited the capillary, the leukocytes move through the tissue guided by secreted cytokines, bacterial and cellular debris, and complement fragments C3 and C5. So the process by which leukocytes migrate in response to a chemical signal is also called chemotaxis. As I said about phagocytosis, that is the most common term that we discussed before in immunology. Basically, during the next and final stage of cellular response, the neutrophils and macrophages, they basically engulf and take in, degrade the bacteria and other cellular debris, any foreign particle, in a process called phagocytosis. So phagocytosis involves three distinct steps, that is the adherence plus opsonization, engulfment, and intracellular killing. Now, intracellular killing happens through enzymes, toxic oxygen and nitrogen products produced by oxygen-dependent metabolic pathways like nitric oxide, peroxynitrites, hydrogen peroxide, and hypochlorous acid. If the antigen is coated with antibody or complements, its adherence is increased because of binding to the complement. So, this process of enhanced binding of an antigen caused by an antibody or complement is called opsonization. So this is the technique. Basically what happens, this is a bacteria which is and you see the opsonin receptors over here and opsonins over here. Because of the opsonin and opsonin receptors, there is an attachment, engulfment, that the cell is engulfing the bacteria, and there is a phagolysosome formation, basically fusion of phagosome and lysosome. Lysosome we know has a lot of hydrolytic enzymes which will degrade the bacteria, and inside the bacteria will undergo intracellular killing. This will also lead to some metabolic changes like protein metabolism is increased, cell destruction, metabolic products leads to increased osmotic pressure in interstitial space, which attracts water and contributes to edema, which is swelling, basically tumor. And the metabolic changes including skeletal muscle catabolism provide amino acids that can be used in the immune system and for tissue repair. We also know about glucose metabolism. Basically, anaerobic utilization of glucose is increased because of hypoxia with increased formation of lactic acid and pyruvic acid. If we talk about lipid metabolism, increased formation of ketones and fatty acids. Mineral metabolism is increased extracellular K plus concentration, and acid-base balance is basically metabolic acidosis. Now we were talking about cytokines which are pro-inflammatory and anti-inflammatory. In case of inflammation, the stage 1 is following an inserted injury. Local cytokine is produced with the goal of inciting an inflammatory response, promoting wound repair and recruitment of the reticular endothelial system. Stage 2 is small quantities of local cytokines are released into circulation to improve the local response, and this leads to growth factor stimulation and the recruitment of macrophages and platelets. And this acute phase response is typically well controlled by a decrease in the pro-inflammatory mediators and by the release of endogenous antagonists, and the goal is basically homeostasis. Stage 3, if homeostasis is not restored, a significant systemic reaction occurs. So the cytokine release leads to destruction rather than protection. A consequence of this is the activation of numerous humoral cascades. Cascades basically means a lot of sequential reactions, and the activation of the reticular endothelial system and subsequent loss of circulatory integrity. So this will ultimately lead to organ dysfunction. If we talk about the systemic manifestations of inflammation, what are the phenotypes? Under optimal conditions, the inflammatory response remains confined to a localized area. In some cases, local injury can result in prominent systemic manifestations as inflammatory mediators are released into the circulation. And the most prominent systemic manifestation of inflammation are the acute phase response, alterations in white blood cell count, leukocytosis or leukopenia, fever, sepsis and septic shock, also called the systemic inflammatory response. They represent the severe systemic manifestations of inflammation. If we talk about the acute phase response, it usually begins within hours or days of the onset of inflammation or infection. Basically, it includes changes in the concentrations of plasma proteins. Liver dramatically increases the synthesis of acute phase proteins such as fibrinogen and C-reactive protein, CRP. In case of any infection, also if you see sometimes blood reports, you'll see the C-reactive protein levels are very high. Okay, because it happens during infection. Basically, increased erythrocyte sedimentation rate, fever. We know fever happens during infection. Increased numbers of leukocytes, skeletal muscle catabolism, and negative nitrogen balance. Now, the acute phase response basically generated after the release of cytokines that we started our conversation with IL-1, IL-6 and TNF alpha. So, these cytokines affect the thermoregulatory center in the hypothalamus to produce fever, right? And IL-1 and other cytokines induce an increase in the number and immaturity of circulating neutrophils by stimulating the production in the bone marrow. Lethargy, which is a common phase of acute phase response when you feel fatigued, it results from the effects of IL-1 and TNF on the central nervous system. Okay. Then if we talk about tissue repair, the primary objective of the healing process is to fill the gap created by the tissue destruction and to restore the structural continuity of the injured part, right? And concomitantly with tissue damage at the peripheral of inflammatory process begins the repair process in order to limit the extension of it. So reparatory processes will involve cell proliferation, conjunctive tissue proliferation, blood vessels new formation, which is also angiogenesis, lymphatic drainage of exudates, and phagocytosis. So injured tissues are repaired by regeneration of parenchymal cells or by connective tissue repair, in which scar tissue is substituted for the parenchymal cells of the injured tissue. So it could lead to malfunction of organs that will also lead to fibrosis, also called fibrosis. Now, chemical mediators and growth factors can orchestrate the entire healing process, facilitate it basically. Some growth factors act as chemoattractants, enhancing the migration of WBCs and fibroblasts to the wound site, and others act as mitogens, causing increased proliferation of cells that participate in the healing process. Like platelet-derived growth factor, which is released from the activated platelets, it attracts white blood cells and acts as a growth factor for blood vessels and fibroblasts. And many of the cytokines discussed function as growth factors that are involved in wound healing. So fibroblasts and vascular endothelial cells begin proliferating to form a specialized type of soft brain granular tissue called granulation tissue. And this tissue serves as the foundation for scar tissue development. It is fragile and bleeds easily because of the newly developed capillary, and the newly formed blood vessels are leaky and allow plasma proteins and white blood cells to leak into the tissues. At approximately the same time, epithelial cells at the margin of the wound, they begin to regenerate and move towards the center of the wound, forming a new surface layer. So as the proliferative phase progresses, there is continued accumulation of collagen and the proliferation of fibroblasts. The collagen synthesis reaches a peak within 5 to 7 days and continues for several weeks depending on the wound site. By the second week, the white blood cells have largely left the area, edema has diminished, and the wound begins to blanch as the small blood vessels become thrombosed and degenerate. Okay. So these are the factors that can affect wound healing. Very briefly, we'll go through it, like malnutrition, protein deficiencies along the inflammatory phase of healing, and impaired fibroblast proliferation. Definitely, collagen and protein matrix synthesis will occur slowly. Then we have angiogenesis and wound remodeling. Carbohydrates are needed as an energy source for WBCs. So fats are essential constituents of cell membranes and are needed for the synthesis of new cells. Then we have vitamins A, C. They have been shown to play an exceptionally essential role in the healing process. Vitamin C is needed for collagen synthesis. Vitamin A functions in stimulating and supporting epithelialization, capillary formation, and the collagen synthesis. The B vitamins are important cofactors in enzymatic reactions that contribute to the wound healing process. And vitamin K basically has a role in bleeding disorders. Basically, it's a blood clotting factor, right? It plays an indirect role in wound healing by preventing bleeding disorders because it will be clotting the blood. Okay. Also, blood flow and oxygen delivery affect wound healing because pre-existing health problems in arterial disease or venous pathology can also affect that. Molecular oxygen is required for collagen synthesis, and it has been shown that even a temporary lack of oxygen can result in the formation of less stable collagen. Okay, we'll not go. Yeah, again, back to inflammation or cytokines, which we started to talk about. So the inflammatory response must be actively dominated when no longer needed to prevent unnecessary bystander damage to tissues. Again, very important. If the response is not needed, it will be unnecessarily harmful for the cells. So failure to do so results in chronic inflammation and cellular destruction. So resolution of inflammation occurs by different mechanisms in different tissues. Meanwhile, mechanisms which serve to terminate inflammation that include short half-life of inflammatory mediators in vivo, production and release of transforming growth factor TGF-beta from macrophages, downregulation of pro-inflammatory molecules such as leukotrienes, upregulation of anti-inflammatory molecules such as interleukin-1 receptor antagonist or the soluble tumor necrosis factor receptor, apoptosis of pro-inflammatory cells, downregulation of receptor activity by high concentrations of ligands. IL-4 and IL-10 are cytokines responsible for decreasing the production of TNF alpha, IL-1, IL-6 and IL-8. Okay. And what about the production of anti-inflammatory repoxins? So evidence now suggests that an active coordinated program of resolution initiates in the first few hours after inflammatory response begins. After entering tissues, granulocytes promote the switch of arachidonic and acid-derived prostaglandins and leukotrienes to lipoxins, which initiate the termination sequence. Neutrophil recruitment thus ceases and programmed death by apoptosis is engaged. Now, these events coincide with the biosynthesis from omega-3 polyunsaturated fatty acids of resolving and protectants, which typically shorten the period of neutrophil infiltration by initiating apoptosis. Consequently, apoptotic neutrophils undergo phagocytosis by macrophages, leading to neutrophil clearance and increase of release of anti-inflammatory and reparative cytokines such as transforming growth factor beta 1. And the anti-inflammatory program ends with the departure of macrophages through the lymphatics. Okay, we'll not go into these. So these are basically a table that I found on the internet. Basically, what are the roles or what are the activities of all these cytokines. I'll upload the slides online so you can go through this table whenever you want. IL, all the interleukins and all the TNF alpha beta, TNF alpha, TGF beta, sorry, all of these, what are their roles? You can figure those out easily. Okay. So we have talked about this. We talked about cytokines, which basically communicate among immune cells. Okay, their proteins. The interaction of cytokine with receptor and target cell can induce a wide variety of responses. They can cause changes in the expression of adhesion molecules. I think we have discussed this. So low molecular weight regulatory proteins or lipoproteins are secreted by WBCs and other various other cells. They assist in regulating the development of immune effector cells. They are referred to as interleukins. IL-1, interleukin-1 through IL-37 have been described. And chemokines, basically they are subpopulation of cytokines that share the specific purpose of mobilizing immune cells from one organ or one part of the organ to another. Okay. Cytokines can be described according to the distance between the secreting cell and the recipient cell. Cytokines that must pass through the bloodstream before reaching the target are referred to as endocrine. Those that act on cells near the secreting cell, such as the cytokine merely has to diffuse a few angstroms to a nearby cell, are referred to as paracrine. And sometimes a cell needs to receive a signal through its own membrane receptors from a cytokine that it itself has secreted. This type of signaling is known as autocrine. So many cytokines act over a short distance in autocrine or paracrine fashion. Okay. Basically, that can be on the basis of the distance between the cell secreting it and the cell receiving that chemical signal. That is the basis of segregation. So, cytokine that induces different biological effects depending on the nature of the target cell is said to have a pleiotropic effect, various activities. Whereas two or more cytokines having the similar function is known as redundancy. Cytokine synergy occurs when the combined effect of two cytokines in cellular activity is greater than the additive effects of the individual cytokines. And in some cases, the effects of one cytokine inhibit or antagonize the effects of another. Cascade induction occurs when the action of one cytokine and one target cell induces that cell to produce one or more additional cytokines. Okay. Each of these forms a network of interaction can be observed in cytokine interferon example. Like interferon gamma causes certain T lymphocytes to secrete additional interferon gamma as well as interleukin 2, and it synergizes with human necrosis factor alpha to activate macrophages and antagonizes interleukin 4 by preventing IL from influencing T lymphocytes. The sensitivity of a target cell to a particular cytokine is determined by the presence of specific cytokine receptors. And cytokines bind to specific receptors on the membranes of target cells, triggering signal transduction pathways that ultimately alter enzyme activity and gene expression. And the susceptibility of a target cell to a particular cytokine is determined by the presence of specific membrane receptors and exhibit very high affinity from one another. This table also will be there. I'll upload the slides. Basically, the total number of protein cytokines and their activities. What are they secreted by all of these? I have already discussed about in detail in one of the previous classes. So I will not go into too much detail. Okay, coming back to the question that we started with in this class. Which of the following are considered pro-inflammatory cytokines? These are the These were the options, and so it is a multiple select question. So the right answers were IL-6, interleukin 6, TNF alpha, IL-1 beta, MCP-1, and GM-CSF, granulocyte mononuclear colony stimulating factor. Okay, so these are the correct options. If you have any doubts, you can ask me. I'll wait for a few minutes and we'll move on to the next question for today. Anyone any question?
These are the sample questions for NPTEL
Yes.
exam, right?
Yes, yes. These are older assignment questions. Yes.
Uh, can you show me the previous questions?
Today we started with this. There was one more question before. And the others were in different classes. Have you not attended the previous classes?
No, no, I'm talking about this class only.
Okay,
That
so
previous
this is
question.
the previous question
Okay.
for today.
Done, ma'am. Thank you.
Yeah. Okay, then we'll move on to the next question for today. 1 second. I'll just go. Yeah. So the next question is, what role does genetic similarity play in syngenic mouse models? So the options are: A. It increases their lifespan, making them ideal for long-term studies. B. It enhances their immune response, providing a model for robust immunity. C. It allows successful cell transplantation without the risk of immune rejection, aiding research on immune cell functions. D. It reduces the sensitivity to pathogens, making them ideal for infection studies. And E. It induces the rapid production of antibodies. And wait for a few minutes. Which one do you think is the correct answer? Anyone? Okay, so the correct answer will be option C. Right? It allows successful cell transplantation without the risk of immune rejection, aiding research on immune cell functions. So, syngeneic mouse models are genetically identical or nearly identical, okay, and they're typically inbred strains. Now, because of this, they share the same MHC or major histocompatibility complex molecules, and the immune system recognizes transplanted cells as self. So, therefore, no immune rejection occurs. This makes them extremely valuable for cell transplantation experiments. Tumor biology studies like tumor grafts are not rejected, and obviously, immunology research where you want to study immune responses without confounding rejection effects. Now, why the others are incorrect? Okay, now if you want to understand the syngenic mouse model, I said they're genetically identical or nearly identical. typical from the same inbred strain. For example, C57BL6 mice transplanted into C57BL6 mice. So they basically means they share the same MHC, major histocompatibility complex, and also the same immune recognition markers. Now, why genetic similarity matters? The immune system primarily distinguishes self versus non-self using these MHC molecules. Now, in genetically different individuals, transplanted cells are recognized as foreign, like immune rejection. In syngenic mice, transplanted cells are recognized as self, so no rejection. Now, what is the core role of genetic similarity? Because of identical genetics, no graft rejection occurs. It allows stable engraftment of transplanted cells or tissues. It enables precise study of immune cell behavior, tumor growth, syngenic tumor models, cell therapies without confounding immune rejection. So this directly supports option C. Now, why all the other options are incorrect? The first option is it increases the lifespan, which is not true. Genetic similarity does not inherently extend lifespan. In fact, inbred strains can sometimes have reduced genetic diversity and health issues. Option B is enhances immune response. Again, not true because these models don't have a stronger immune system. They have a controlled, consistent one which is different. Option D reduces sensitivity to pathogens. Again, not true because many inbred mice are actually more susceptible to certain infections. Then, the option E is induces rapid antibody production. Again, not correct because antibody production depends on immune activation, not genetic similarity per se. Okay. So, if we start talking about model organisms for the first time. So, what are model organisms and basically how does it help our research? A model organism is a species that has been widely studied, usually because it is easy to maintain and breed in a laboratory setting that has particular experimental advantages. Now, model organisms are non-human species that are used in the lab to help scientists understand biological processes, right? For example, they may have particularly robust embryos that are easily studied and manipulated in the lab. This is useful for scientists studying development. Or they may occupy a pivotal position in the evolutionary tree. This is useful for scientists studying evolution. Now, why are model organisms useful in genetics research? So, many model organisms can breed in large numbers. Some have very short generation time, which is the time between being born and being able to reproduce. So, several generations can be followed at once. So, mutants allow scientists to study certain characteristics of diseases, and these are model organisms that have undergone a change of mutation or mutation. So, in their DNA, that may result in a change in certain characteristic. Now, why are model organisms useful in genetics research? Some model organisms have similar genes or similar sized genomes to humans. Model organisms can be used to create highly detailed genetic maps, which are basically nothing but visual representation of the location of different genes on a chromosome. A bit like a real map, but one where the key landmarks are areas of interest in the genome. For example, areas of DNA that differ between individuals in the same species, like single nucleotide polymorphism or genes. These are some of the examples of model organisms that are used to study genetics, like yeast, Saccharomyces cerevisiae, fruit fly, Drosophila melanogaster, nematode worm, Caenorhabditis elegans, western clawed frog like Xenopus tropicalis, then mouse, mus musculus, zebrafish, Dinorario, monkey, obviously Macaca mulata. One second, some issue in the Google Meet. Yeah, one second. I thought sample types. Okay. Yeah. Now, what are the examples of model algorithm used to study genetics? For example, Drosophila, the reason low cost, rapid generation time, small genome, used to study each functions of genes, genetic mutations. Zebrafish also low cost, rapid generation time. Embryos are transparent, so microscopy can be done easily. Easy genetic engineering used to study developmental diseases. Xenopus eggs are easily manipulatable. Eggs are fertilized as the animal used to study embryonic development. Rabbit again, short life cycle, very docile, easy to handle, phylogenetically close to human, used to study all kinds of diseases. And rodents, the most used animal model, easy access, possibility to modify genetically, very similar to humans, and they are used to study all kinds of diseases. Macaque or monkey, they're genetically very similar to humans, right? Similar immune system to humans, useful for longitudinal studies. So these are some of the examples. 2D, 3D Homo sapiens cell culture starting from, and these are all the okay. So what are the things that are done? Precision medicine, identification of therapeutic targets and drug testing, forward mutagenic strains, and validation of variants pathogenicity. Okay. Now, over the past century, the house mouse, Mus musculus, has become the preferred mammalian model for genetic research. So, in the early days of biomedical research, scientists developed mouse models by selecting and breeding specific mice to produce offspring with certain desired characteristics. Now, scientists use mice to simulate human genetic disorders in order to study their development and test new therapies. As a scientific tool, mice have helped to speed up the progress of research and enabled the development of important new drugs. They have many similarities to humans in terms of anatomy, physiology, and genetics. They have similar genome, making mouse genetic research particularly useful for the study of human diseases. They are small and cost effective. They are cheap and easy to look after. They multiply quickly, reproduce as often as every 3 weeks, creating lots of offspring. Okay, generation time is short, around 10 weeks. Several generations can be observed at once. They have a short lifespan. One mouse year equals about 30 human years, which means scientists can easily measure the effects of aging. They're extremely useful for studying complex diseases such as atherosclerosis and hypertension, as many of the genes responsible for these diseases are shared between mice and humans. So, research in mice provides insights into the genetic risk factors for these diseases in the human population. It's relatively easy to manipulate the mouse genome. For example, adding or removing a gene to better understand its role in the body. This provides a powerful tool for modeling specific diseases when a mutated gene is known to play a role in the disease. Now, mice are far better than flies or worms for studying complex biological processes found in humans, such as the immune, endocrine, nervous, cardiovascular, and skeletal systems. Like humans, mice naturally develop diseases that affect these systems, including cancer and diabetes. Immunodeficient mice without a fully functional immune system, can also be used as hosts to grow both normal and diseased human tissue. This has been a useful tool in cancer and AIDS research. So, basically, genetically modified mouse is a mouse that has had its genetic genome basically altered through the use of genetic engineering techniques, right? And genetically modified mice are commonly used for research or as animal models for human diseases, and they're also used for research on genes, transgenic mice. Examples of mouse models like patient-derived xenografts are models of cancer where the tissue or the cells from a patient's tumor are implanted into an immunodeficient or humanized mouse. Okay, will not go. So why use transgenic mouse models? So model specific effects of human disease. They can test drugs or other treatments in a control system. Then highlight the function of a gene of interest. They can cross to different lines to expand experimental possibilities. If we talk about transgenic and knockout mice, a transgenic animal is a much more complex than working with cultured cells. Transgenics can be used for a variety of purposes, covering both basic research and biotechnological applications. Now, mice have 19 autosomal chromosomes and the two sex chromosomes X and Y, many of which contain large segments of DNA that are highly conserved between mouse and humans. Okay. Now, one of the goals of mouse transgenesis is to use molecular genetic approaches to create better mouse models of human diseases that are based on known genetic lesions. And the mouse is a reliable stand-in for humans in medical research thanks to genome that is 85% identical to us. Okay. If you talk about synteny, that's basically the colocalization of genes on chromosomes of related species. Homolog, basically the situation where nucleic acid or protein sequences are similar because they have a common evolutionary origin, often used loosely to indicate that sequences are very similar. Ortholog gene sequences are similar between species, and paralog gene sequences are similar within a species. Okay, you see transgenic mice where the hair gene has been removed. Okay, used to help burn patients and others making human facial parts. Comparison between the transgenic mice and a normal mouse. Okay, the giant mouse developed from a fertilized egg to transform to the recombinant DNA molecule containing the human growth hormone. Then we can knock out the expression of NHLH2, a basic helix loop helix transcription factor in mice, results in adult onset obesity. If you see these differences. Okay, so these are some of the differences. They can be spontaneous mutants, like occurs as a result of spontaneous mutation. Examples can be DBDB, diabetic mouse, nude mouse. They can be induced mutants, transgenic, overexpression, inducible, conditional, gene targeting, knockouts and knock-ins. Mutagenic mice, basically chemical mutagenesis of embryonic stem cells, irradiation of mice. So what is the step of creating this? Step 1 is inject DNA construct into zygotes. Implant transgenic zygotes into pseudo-pregnant females. Step 3, genotype founders. Then transfer to pseudo-pregnant female. Obtain germline transmission from chimera to establish a new line. Okay. So basically, if you modify a region of the DNA and also homologous recombination in embryonic stem cells, they can be injected into E cells in the blastocyst, transferred to pseudo-pregnant female, and which is obtaining a germline transmission from chimera to establish a new line. A pronucleus is the nucleus of a sperm or an egg cell during the process of fertilization, right? So the sperm cell becomes the pronucleus after the sperm enters the ovum, but before the genetic material of the sperm and the egg fuse. The objective is to alter the germline so that the genetic chain is inherited in a stable pattern. Now, transgenes should include promoter with or without enhancer, gene to be expressed, which may be a reporter gene, splice donor or acceptor sequences flanking and infron. These can be from another gene like beta globin or SV40 antigen. Termination polyannihilation sequences. These can be from another gene, beta globin or SV42. Now, transgenes must be excised from the bacterial plasmid sequences in order to be expressed in mice. This is because the prokaryotic cloning factor vector sequences inhibit expression of eukaryotic genes introduced into the mouse genome. Basically, this sequence is ensuring the expression of the transgene. Okay, I'm not going into. Okay, now what's a knockout mice? A knockout mice is a mouse in which a gene has been deleted or mutated. Gene is inactivated. Specific gene is targeted. The loss of gene activity often causes changes in a mouse's phenotype, and this provides valuable information on the function of the gene. Embryonic stem cells are stem cells derived from the inner cell mass of an early stage embryo known as the blastocyst. Embryos reach the blastocyst stage 4 to 5 days post fertilization. They consist of 50 to 150 cells. Now, ES cells are pluripotent, are able to differentiate into all derivatives of the three primary germ layers: ectoderm, endoderm, and mesoderm. So, these include each other more than 220 cell types in the adult body. Now, targeting construct for positive negative selection. Now, how to make the targeting construct? A positive selectable marker flanked by two arms of homologous sequence and negative selectable marker outside of one homologous sequence. So positive selectable markers are basically expression cassettes encoding antibiotic resistant gene like neomycin. Okay, it's an antibiotic resistant gene, so it's a positive selection cassette. Now, what is the selection strategy? So, positive selection is basically G418 neomycin resistance gene. It confers resistance to G418 antibiotic. Now, negative selection is ganciclovir, herpes simplex virus thymidine kinase, HSV-TK gene. It's sensitive to ganciclovir, selects against random integrants. Okay. Now, basically, you generate the CK targeting vector, electroporate the vector into the ES stem cells, basically oocytes of the zygote, then culture ES cells in the presence of selected and peak resistant colonies, identify the ES cell clones with desired targeting either by southern blot analysis or long range PCR. Microinject targeted ES cell clones into blastocysts and transplant into uterus of pseudopregnant female mice. Basically, they can identify chimeric pups and generate progeny that can carry the targeted allele. If you see gene targeting, the embryonic stem cells are cultivated from mouse pre-implantation embryo blastocysts. In the construction of the targeting vector, the vector contains pieces of DNA that are homologous to the target gene as well as inserted DNA which changes the target gene and allows for positive negative selection. The ES cell transfection, the cellular machinery for homologous recombination, allows the targeting vector enables the target vector to find and recombine with the target gene, and proliferation of targeted ES cell selection for the presence of neo and absence of HSV-TK enriches targeted ES cells. Now, from gene targeted ES cells to gene targeted mice, after this, if you see, basically the injection of ES cells into the blastocyst, targeted ES cells are injected into the blastocyst, the mosaic inner cell mass, if you can see over here, where they mix and form a mosaic with the cells of the inner cell mass from which the embryo develops. Then implantation of the blastocysts, the injected blastocysts are implanted into surrogate pseudopregnant female mother where they develop into chimeric embryos. Then birth and breeding of the chimeric mice. The chimeric mice mate with normal mice produce gene targeted as well as normal offspring. And then we see birth of gene targeted mice. Gene targeted mice called knockout mice when the targeted gene is inactivated in all the cells. We can do conditional knockout, we can do knock-in or additional gene function is established, we can do laxizine as a means. So in 1995, a database was established to collect details of knockout mice. It's known as the mouse knockout and mutation database. There are over 5,000 entries in the mouse knockout and mutation database, which is a major resource for those interested in using mouse model as a system for the study of gene expression. Okay, you can go check this out. Now, if you talk about conditional mutagenesis, okay, so you want your target gene is knocked out in all tissues in all types. This is a conventional knockout. But if you want to do in a condition, you control where and when spatiotemporal selection of where your targeted gene is knocked out. So in this case, the Krelok's recombination system comes into play. The Krelok system is used as a genetic tool to control site-specific recombination events in genomic DNA. The system has allowed researchers to manipulate a variety of genetically modified organisms to control gene expression, delete undesired DNA sequences, and modify chromosome architecture. Okay. So basically what happens, the targeting vector contains three LOXP sites that flank the regions of the genes of interest to be removed and the positive selectable marker mu. After homologous recombination, the selectable marker is excised and transient expression of CRE occurs, CRE protein 3, and the correct recombination is identified by screening this of the analysis by its or by PCR, long range PCR, and the mutant ES cells are then used to produce mice. A subtle mutation is introduced along with the selectable marker in the targeting vector, right? As selectable marker is then removed by transient expression of SCRE, which leaves only the subtle mutation in the small logs besides the silent location. A transgenic animal which is finally bred to an animal line expressing the Cree under either a temporal or spatial control. So Cree can also be ubiquitously expressed to obtain a knockout in all tissues including the germline. Okay, these are the strategies. Not going into too much detail. I will upload the slides, you can go through it. I'm not going into too much details. The most widely used mouse strains are C57BL6 and BALB-C mice. Nomenclature for mouse strains, you can check out this website as well. Coming back to the question that we started with today, what role does genetic similarity play in syngenic mouse models? The answer will be it allows successful cell transplantation without the risk of immune rejection, aiding research on immune cell functions. Okay, so this question is done. If anybody has any queries regarding this question, any doubts, you can ask me now. I'll wait for a few minutes. Okay, so if nobody has any question regarding this, we can stop today's session here and we'll meet again tomorrow for the extra session. I will upload all these slides. I'll upload all these slides online and we can yeah, sure. Tomorrow is the extra session. We can do the revision of those. Yeah, for sure. So I'll conduct the session. I'll stop the session now, and we'll meet again tomorrow. Thank you.